Hello and welcome to Archaeology 101, and in today's topic I'll be discussing how archaeologists identify disease in the past by using genetic analysis. So let's begin with some background on disease and the human skeleton. Disease can impact the human skeleton, but this depends on a few factors such as host resistance, wherein the host has to survive long enough for the disease to remodel the human skeleton. Any remodeling of the human skeleton caused by disease is known as pathology. This pathology can be very hard to distinguish between diseases. One pathology may represent two different diseases. They both represent very similar types of pathology. Also, this is unaided by the limitations of how the bone reacts. However, there is a few examples where you can distinguish different diseases. If you look on the bottom left hand of the screen, there is a spine which has displayed massive remodeling caused by tuberculosis. This is known as Pott's disease, and it's pathognomonic to TB, meaning it's usually specific to this disease. On the other hand, you have non-pathognomonic pathology such as periostitis, which you can see on the bottom right hand corner, and you can't use that to identify one particular disease because it occurs across a whole spectrum. Now, in certain cases, it is possible to use genetics to study ancient disease. A classic example, and one which is quite relevant to today, is the 1918 Spanish flu epidemic. The analysis conducted was using soft tissue extracted from well-preserved historical examples from an Inuit lady who is preserved in permafrost and World War I veterans. From this analysis, they were able to identify that it was the H1N1 virus, which had probably jumped from birds to humans. However, not all examples are so well preserved, and this is a historical example. More ancient examples do not have soft tissue, meaning it's harder for ancient disease to be preserved. The process of studying ancient DNA, or aDNA as it's known, is usually used through a process called polymerase chain reaction, or PCR, although you can use other techniques which take this principle, such as whole genome sequencing. Essentially, you just remove a sample of human bone, which is a destructive sampling technique, turn this into a solution, which you can then put through the PCR process, where you can amplify for particular DNA strands, some of which might turn out to have been ancient disease. In archaeology, aDNA has had three main successes in studying aDNA of disease. So that's tuberculosis, Mycobacterium tuberculosis, leprosy, Mycobacterium leprae, and bubonic plague, Yersinia pestis. These are because they have a harder external protein membranes which aid in their preservation, whereas other similar diseases may not have this protection and they may not survive in the archaeological record. TB is very hardy, and originally it was thought that it may have interacted with humans by around the Neolithic period and had jumped the species barrier from animals such as cattle when we started engaging with animals much more closely. However, aDNA analysis shows that human species have had TB for about 3 million years, and this radically changed our perception of TB. This is a very old relationship that we have with this disease. The success of aDNA is partially due to commercial interests, because you can use it to study disease and you can use it for food. So it's just increased in its research, and there's much more to come out of aDNA, and many people have heralded it as this all-powerful technique. Undoubtedly, aDNA is a really powerful tool. However, I'm going to rain on the parade here and show you the limitations as well, which aren't really portrayed in the media. So there's research bias to begin with. In archaeology, osteologists will often get specimens which already display pathology, so we already know there's something wrong with the individual, and they will usually use aDNA to only identify the disease of individuals which display pathology. But we've already noted that pathology is not present in every single sample who may have been infected with the disease. Secondly, there's preservation issues. So aDNA cannot look for every single disease, it seems. Syphilis has been a major crux in being able to be identified by aDNA because the microbes, the spirochetes, which they're known as, are very vulnerable to things such as sunlight, and they just disintegrate. Thirdly, there's contamination. There's quite a funny but sad story, I guess, of dinosaur DNA. This is when paleontologists were really excited in the earlier days of aDNA and thinking, oh, we can use this technique to find DNA of dinosaurs. Supposedly they found dinosaur DNA, but then someone pooped on their parade and found out, oh, this is actually human DNA all this time. They had been looking at their own skin, essentially. There are also many other issues, such as different types of contamination, which include bacterial, fungal, and transport contamination processes as well, which completely mucks up the entire process. So let's recap. aDNA has an excellent potential when used in the right conditions. Clearly, TB has been really well researched when using aDNA because it can. And there is much more to come from aDNA as there is commercial interest in the process. So that's why it's so rapid in getting better and better, and it will continue getting better as well. However, it's still not this all-powerful end-all be-all technique which some people such as David Reich have perpetrated. It's very vulnerable to contamination, unfavorable preservation, and also there are research biases when using it, which one has to always keep in mind when looking at genetics. I'd like to say thank you very much for watching, and I hope to see you next time on Archaeology 101.